Hey Jolkson, how's it going? I saw you streaming earlier today. I'm used to you streaming for like hours and hours. You... You're just on for a while, yeah. These are some prepared sample from a small pond. Four samples from this pond. Slides from another pond. Woods that haven't looked at this yet. Stream anywhere. Wednesday I'll probably look at some. Particularly interested in this sample pond that I grew up swimming in. Hey Pacific. I was a little baby kid and learning how to swim. Would be owned by. He passed away. Kid. Sold off the property. They kind of chopped it up. This went to my. Or she was inherited maybe some. Pond that we swam on went to someone else who bought the property. Mom's house, just living a little bit from where. Oh, the mic is cut. Or what that's about. Yeah. There. That should get rid of that. I think it's just the noise gate was acting a little weird. Is it still cutting out? Maybe there's something wrong with the microphone. No, okay, good. Yeah, the noise gate was for, I think, when we had a fish tank in here. And sometimes when I'm running the fan. To try to keep it from doing that weird thing. Anyway, so that's sort of a long story about the sample. The pond looks like this today. This is not the way this pond looked when I was a kid. But my uncle's house was out here somewhere. Oops, that picture's not somehow in front of my arrow. Give me a second. Making some adjustments. There. Oh, my uncle's house was somewhere over here, and we walked up some little trail to get to it. And then, we jump in off of this dock or whatever, swim out in this pond. It looked a lot different back then. These people maintain it a lot differently. They actually mow right up to the edge of the pond and had cattails and stuff in it when I was a kid. So I remember playing with those, but there's no cattails anymore. And there's something weird going on with the property. It's like there's a fence right here and another fence right here. And it's like the people who bought this property wanted to have access to the pond or something. So they blocked it off here and here as their property, and then the people who own it now, this is the property, I think. At least I think that's what's going on. Anyway, my mom and I went up to this pond when I was home, and we took some pictures of dragonflies that were all around this outside edge, and then some pictures of barn swallows, I think, that were on this little fence edge here. And the people who own the house are, like their neighbors and friends with my mom. So they saw a bunch of the photos on Facebook, and they'll probably watch this stream, which will be kind of cool. Anyway, we came back a couple of days later and asked them if it was okay if we took some samples from the pond. And I snuck up here and threw a ice cream bucket with nothing, an old ice cream bucket with nothing in it, tied to a rope, and pulled some plankton samples in from here, off of the dock. And then we collected some material from here, from sediment from over here. And then on the other side of the pond, there was a little, another little area where we collected some materials. So those are samples that we got from this. And the plankton, mostly they all have the same thing in them. So it doesn't really matter that much about where we look at the sample. So I just picked one of them. Was that Calanese? I don't know what you mean by was. Do you mean, is this Calanese? Is. No. This is a Nominese. It has this like landing strip of areolae along the raphe on both sides. The runway, yeah. And actually, this is very similar to the Anomalies that we saw on the stream today from the SCM. I don't know what the, I guess it's Sphaerophora probably. Because Costata looks more like a football, and Sculpta, maybe it's Sculpata or Sculpta or something like that. I thought has a little bit of a capitate head, but I don't remember off the top of my head which one it is. They're all forms of a similar thing. Yeah, you could probably click in there. They might actually have this one posted. Maybe I should click in there. Yeah, it's the one right there in the picture, I think, with the 1 and the 2 on it, and probably the 3 in that guide. So that's pretty cool. But this is it. And I don't actually care that much about this thing. This is something everybody knows and has seen a bunch of times, but this thing's kind of interesting to me. But the Anamnes is really pretty, so I thought maybe we would just take a look at it real quick. And we can, oops, that's the wrong button. Can zoom in and look at some of the detail. The Raphy ends have these sort of really cool button hook ends on them. And then there's a sort of runway or whatever, with lights all along the edge. And then these things are called ghost areolae. Sculpta, yeah. These things are called ghost areolae, so they don't actually go all the way through the valve, but these ones do. So they look like little shadow bubbles. They're really kind of cool. And then the striae continue, they go from being punctate into these sort of more defined lines near the edges. So it's kind of cool. And we can look very closely. So we're at 1,000 times magnification, multiplied times 3, so like 3,000 times magnification. This is as zoomed in as we can get. And I don't actually have a scale bar for 3 times 1,000, but that's what it is for 1,000. Right there, that's 1,000 times magnification scale bar. So this thing is maybe 80 or 90 microns in length. It's a pretty big diatom. And I think it's really pretty, right? So get a good sense of it, kind of lemon shaped or hamburger shaped. And then this guy is actually the one I wanted to focus on. I saw this and I was like, I don't know what that is. And I'm still not totally sure what it is, which is pretty interesting for a round diatom. Because most of the round diatoms, I can get them into genus in the freshwater pretty easily. There aren't that many freshwater genera, centric diatoms that are like this. But I think maybe it's what was called or is called Spicatacribra, but I'm not positive about that ID. This one doesn't look to have a lot of bigger areolae in the center like Spicatacribra always does, but some of them that we've seen definitely do have it. So I'm a little torn as to what it is. It's kind of domed. You can see both valves. That's the back valve, that's the top valve. And there's no real structure. It's just like, radiating rows. And some of the ones where there's only one valve, you can actually see the mantle photoportula. You can kind of see them here just barely as shadows, these dark blotches. But I'll see if I can zoom around and find one that's you can kind of see it when I zoom out as well. That has the just one valve, so you can kind of see those. And these little guys that we're driving past right here that are in colonies are probably Pseudosaurosyra brevistriata. This one up here definitely is. Oh, sorry, down here, it's upside down. That one definitely is. These ones I'm not positive about. They could be a Staurosyra. Here's another one where the central areolae are a little bit bigger. Let's see. Still not huge. You can see the strutted processes and rimoportula are out here on the outside edge. So I didn't know what it was, and I actually asked my wife Carlin, and she also didn't know what it was. So we stared at that for a little while, and then we looked at the different descriptions and kind of came to the conclusion that it's probably Spicatacribra, but we're not sure. And the SEM will really help with that. It's an example of how light microscope stuff can only get you so far. This is Platessa zeigleri. So that's a monoraphid diatom that I see all the time. Really common. That is a chain of Starosera construens. And there's a little guy here that's some sort of acanthidium, but I'm not positive which one. That is a eunotia. Some more Brevistriata. There's another one where you can really see the strutted processes quite well, because it's just a single valve. And that one really starts to look like it has that sort of bubbly center of Spicatacrubra. It's a lot more obvious when you're just looking at one. I think that might be because there's some interference. But you can also see the strutted processes are here, and I'm gonna guess that's a Rimiportula because of the spacing. These are all pretty evenly spaced around like a clock, and then there's one that doesn't look like it matches. So that's just a little diatom intuition that tells me that's probably the Rimiportula, because these ones are all basically otherwise evenly spaced, and it should have a Rimiportula. So, I mean, that pretty much looks like a well-behaved Spicatacrubra. So I'm not sure, but that's where I'm putting it for now. And I was really excited about that because I've never seen one. And I was particularly excited because I've never seen one, and it's in a lake that I grew up swimming in. So I choked on some of this water when I was a kid. I'm sure of it. Playing and doing whatever else, which means some of those are probably stuck deep in my bones, which is what happens with little tiny diatoms. There's a Nitzia. Hopefully there's no Nitzia being carried around by me. But you can see that little tiny guy, the little Spicatacrubra, is actually really quite common and can be a little bit bigger. There's one. I'm just going to guess that that's what it is for now, and then when we get on the SEM, I guess I can disprove it or prove it one way or the other. And that little guy right there is a Discostella. This one, that's not the same thing. That's clearly a Discostella. Hey, Steve. They get stuck in your bones, yeah. What happens is you can see it's a Discostella. There's the central little flower structure. And then it's really tiny, so you can't really see where the Rimiportula is, but I promise that's a Discostella. It's either Styligia or Pseudostyligia or something in that group. Steve, what happens is you choke on it, so it goes into your lungs, and then your lungs have these little openings, these alveoli, these little openings where the oxygen is exchanged with the blood, and the holes that are there are big enough that silt-sized particles can actually get into the holes, which means they get into your blood, and then it goes around in your circulation for a while. And when your body is trying to purify the blood, what it does is it dumps the waste material that it finds in it into the marrow in your bones. And so, yeah, they get into your bones, basically. And that's where they stay. Because once they're there, there's no coming out. So, any diatoms that you've choked on in your life that made it into your lungs very likely went into your bones. And you're probably carrying them around as debris or junk, which is kind of cool. We're all probably carrying around some. It's a piece of a Nitzia. Somewhere over here I found, oh, that's a big one. That one looks like an initial valve up here. It's very round on the back side. What's cool about that is that's pretty small for an initial valve. It's like maybe 15 or 20 microns. There's a little navicula. Somewhere over here I saw a giant Cerarella. There's a bunch of them, and I may need to actually jump out to a lower magnification in order to find it, because it was really kind of cool. I thought it was on the same sort of general line that I was on. Eunosha. I'm about to the edge of the slide, so I think I'm gonna just jump out for a second and see if I can find it at a lower magnification so we can take a look at that. Yeah, earlier I had some really cool stuff on the SEM. I think Steve saw the pictures on Facebook that I posted. Those were, that arachnodiscus was from some material that Pac sent me, and I have it here as prepared slides as well. So we'll take a look at it as soon as I find this. Well, I think I'm going to look around a little bit more in these slides, but soon we'll jump over and take a look at some of that stuff from the marine realm. Oh, there they are. So also I found these really big Cerulella. This is them. And I don't recognize them, which doesn't mean a lot, because there's probably a ton of Cerulella that I don't know, but like never seen before anything like it. So thought it would be cool to take a look at it. See, it's mostly like flat in the center. Get a sense of how big these things are. That skill bar is correct. So this thing is probably 150 microns, which is basically the limit of my viewing on my microscope. So yeah, those were the colorized images that I put up where the arachnodiscus. So if you like that story, Soph, about diatoms being trapped in your bones, there's a bunch of cool stories that are related to it. One is that people use diatoms for forensic analysis. You'll sometimes see them on CSI or Dexter, not the cartoon Dexter, but the Showtime show Dexter. They almost caught him because they traced the diatoms from the trash bag rocks that he was using to bury the bodies at the sea back to a specific pier through the diatoms, which is probably unlikely, but people have used pollen and diatoms to track whether something is a marine, whether a body's been moved after it's been drowned, or to trace people, like, you know, they find some mud in their car tires and realize it's lake mud. So if you're into forensic stuff, I think there's some forensic files or whatever where they just talk about diatoms a little bit. They probably butcher all the names. But, and I have a friend who is like a Twitter friend who does forensic diatom work. I should have her on. I should see if she could come on my stream and talk a little bit, do an interview with her, because people seem really fascinated by the use of diatoms in forensic science when I talk about it. It's not something I've ever done, so yeah, I don't recognize this thing, and I still don't recognize it, and it doesn't look like anything that's on Diatoms of North America, and so I'll have to look through my books and see if I can find something like this. But it's really curious because the center is all basically clear. There's striae there, but you can't really see them. Yeah. So, yeah, they use them for forensic stuff. I can look her up and maybe put a link to her Twitter in Discord, because I don't know what her handle is off the top of my head, and I don't want to spend time digging through my Twitter looking for her account. Yeah, it's like a real skinny little guy, and it's in many of the samples. So there's that little Spicatacribra. There's another one like right, I don't know where, oh, right here, right next to it. You can see. So I'm kind of curious what that is. But they're both same Sorella. So it'll be fun to dig around and look at that on the SEM as well. So that should be available. Should be something that we find pretty easily in the samples on the SEM. I was pretty close to it when I had that giant Nitzia. Anyway, so diatoms we've seen, Spicatacribra, potentially, giant Sorellas, a couple of different kinds actually. There's another one in here that's a pretty common, let's see, pretty common Sorella. Oh, Tenera, I think, is the one that's in here. And also, I think Tenera was the one that we saw in the Mountain Lake samples from that Pacific Plankton gave us. Yeah, Spicatacribra is on there. And if you click on that link, I can actually show it. Let me put one of these back on here that has a really distinct central area. And then there's a Nitzia. That's a Eunotia right there. Sort of moon-shaped one, or sort of bent. Whenever you're not looking for something, then you find a bunch of it, vice versa. There was one, but it didn't have the central. I don't know. It's not consistent, whatever that is. It's a Nitzia. Oh, there's another giant anemones. It's a bunch of these. Let's look at this one, because this one's got this sort of central bubble. And then, like, the center area in here is a little bit bigger areolae than the rest of it. It's maybe slightly domed. Anyway, so that's, let's see. Oh, do I have to have this open? Yeah, okay. So this is Spicatacribra, and when they're really little, they're like this. It's not totally clear they always have that sort of central bubble thing, and this is what it looks like in SEM, so, you know, we ought to be able to see if that's the case. Pretty interesting little guy. Bunch of Discostella, Salafra. Also on the stream earlier, we saw star anise. That's a star anise. I don't think it was this one that we saw, but something similar. The central part of it right here is a big clear section with no striae on it. All the way around to the edges, no striae. And that's characteristic. This feature is called a staros on diatoms, on this diatom. And inside there's a sort of thickened silica section for that. that sort of matches it. Yeah, look at you. Working on the spelling. This is the raphe. These are the striae, and they're made up of rows of puncticulate or little dots, basically. That's characteristic, the combination of that central stripe plus the rows of dots that are separated into little, or the striae that are separated into little dots. Pretty much how you can tell that you're looking at a star anise. It's a cool diatom. Here's a whole bunch of colonies. It looks like Starocira and Pseudostarocira colonies. Those look like there may be Fragillaria. There's a Brevistriata. No, Brachycira. Sorry. Another Navicula. Anyhow, it's pretty much the assemblage that we see in the lake. These little tiny guys, some Knitsia, some colonial stuff. If I were looking at this sample, I don't know anything about Spicatacrobra, where it's found. Little Pond is not where I would have guessed it was found, but it is small. Oh, there's a Gomphanema. It's pretty cool. It's asymmetric. Oops, I pushed the wrong button. Let's see if we zoom in. You can kind of see some of the detail. Prepared slides don't have the same kind of excitement as when we look at live material, but the detail is much better, and you can see all of the cool things. Like there's a stigma, here's a isolated striae in the center area. There's another one over here that's shortened, and then this diatom is asymmetric. What I mean is, if you were making paper snowflakes, you could fold it along this axis and then cut a pattern and it would make the diatom. But if you fold it the other way across this direction, they don't match up. So it's only symmetrical and along the long axis. And that characteristic will get you pretty close to Gomphanema and most of its allies pretty quickly. So a few of its allies are slightly outside of that description, but it'll get you close. There's a bunch of these little guys. They're everywhere in the samples, so that's nice. We won't have any trouble finding it in the SEM. And then there's Eunotia. So I would guess this is slightly boggy, acidic. I mean, I took these from plants, so, or near plants. So I would guess that that's just some sort of like plants are close by. There's a interesting thing that's what's the name of that thing? Ctenophora. That's what it looks like anyway. Starts with a C, not a T. Anyway, I'm going to look around a little bit more and then we'll jump over to the samples from my mom's pond. Like I said, I'm going to send this link over to my mom's friend and my mom so they can check them out. So now's your chance to say hi to Dia Mom if you'd like, because she'll probably see it. I don't know if she'll read the chat. She might just look at the pictures. Yeah, that's definitely Ctenophora right there. You can see the same structure. There's another Sororella. That's like Libriel. Really, the stream is just showing my mom what I found in her pond and her friend, in a way, at least the first part of it here. And then we'll take a look at some of the stuff that Pacific Plankton sent me that we saw on the SEM today. It's kind of nice to have both of them together where you can kind of look at what we saw in the light microscope and then or rather in the SEM and then see it in the light microscope. And for my students, that's actually what I think is really one of the most productive things, is being able to bounce back and forth between them so they can kind of see all the detail in the SEM. That's a Sir Salaphora right there. Little tiny Salaphora. She's probably not here. I don't think my mom has Twitch or follows me, but as she watches the links, and she watched my bird link. I caught her. Well, it was the birds that were at her house. She just wanted to like see what I found, I think, and she was really interested in some of the birds I saw. I mean, she's seen them all before, but I got some nice close-up images of them. There's a Pinularia. It's a long Gomphanema right there. I haven't seen any in foul view that are like that. It's Eunotia and Girdle view. Little guys, a bunch of little guys growing on things, or they were. These samples I did these samples today. I digested them in nitric acid, and when I thought that it wasn't going fast enough, I put a little bit of hydrogen peroxide in it as well. So there's a Nidium, Navicula. So it sort of sped up the reaction a little bit, and then I simultaneously was making SEM stubs, prepping these samples, and prepping Pacific Plankton samples all together. So sort of juggling all three of those this morning, and then looked through some of them just briefly to see what was in them, and then stuck them on the SEM. So there's the Eunotia again. A nice little acid bath, yes. Hey, Commander Shafard. Cabbage. Alright, I'm going to switch us out. Let's see, with one of the samples from the pond behind my mom's house. So some of these samples were collected with they were mud, and some of them I sort of skimmed the surface. I think maybe this one is a good one, but we'll see. Like I said, a lot of the samples had basically the same thing in them. They're ponds, so they're quite small, and there is a diversity of habitats, but it's sort of like mud, near shore, plants, pretty much all benthic habitats. And probably if I look through them, I'll actually be able to see some of the differences in them. But for now, they're basically all pretty similar. At least in bulk, I was pretty consistently seeing the same things in those samples that we saw. We saw most of it. This sample, as I mentioned, is from a different environment. This is from a wooded pond behind my mom's house, and sort of an adventure collecting this sample because there's no access to the actual water, really. Yes. My mom's pond is definitely stagnant, and the neighbor's pond is basically well maintained and It used to have a standpipe in it, so I think it is probably aerated. So it doesn't have any like clear algal blooms. You can actually see to the bottom from the dock pretty much. In contrast, the one that we're about to look at, so I'll bounce back and forth between them once I get this image in focus here. Oh, that's a pretty little diatom. Symboplura. Let's see, for pragmatic, this is the pond we were looking at. It's pretty well aerated, that I was talking about I swam in. And this is the one that's in the forested area, my mom's house. You can see there's like no part of the pond that has open water whatsoever. And I had to put down a piece of wood out into the water here because it's really shallow, but I couldn't, you can see this is all mud basically in here. And then so maybe it gets a little deeper and turns into sort of water here, but if I stepped into that, I probably would lose my shoe, and who knows what else. And then there was just dragonflies all over the surface. This is all duckweed. The entire surface is covered with duckweed. So this is definitely a dystrophic, anaerobic environment. There's no light getting to the bottom. It's all blocked off by the duckweed, and so you only have plants in these little, marginal areas, which I think is pretty cool. And I was just sort of curious. I never swam in this one. I never actually even went out on a little boat or anything on it, but there was a boat nearby, like somebody had been doing that. But it didn't look like anything anybody had used recently. So really shallow. I didn't find much in the way of plankton in this lake, but I did find some cool diatoms. So here's this Symboplura, as I mentioned. You can see there's a bunch of rocks and stuff or sand grains and silt grains, because it was collected more or less from areas where there was mud. That is a cnidium, and you can tell that it's a cnidium because the raphes in the central area go hooking in opposite direction. So the proximal raphe, which is this one, hooks in the opposite direction. And there was a whole really cool Diatom Web Academy talk by Paul Hamilton, where he talked about all the different cnidiums. And probably, I think Pacific Plankton saw that talk. I watched that one. It was really great. And Paul's a fun person to listen to. You should definitely check it out. If you're interested in diatom taxonomy, that's primarily what it was about. Here's a Pinularia. So Pinularia are pretty easily identified if you know what you're looking for, which is, oh no, paleontologizing with a giant raid. Thank you, paleontologizing. You brought a bunch of dinosaur people over to look at my diatoms again.
Good news, everyone!
That. We're looking at some fixed samples. I know a lot of times I'm looking at like stuff that's moving around.
Good news, everyone!
Thank you for all the follows. This is a pond from behind my mom's house. I went home. I turned 50 last week, and I went home to visit my family, and
nothing is impossible.
And
Not
hung
if
out
you
with
can
them
imagine.
for my birthday. And then at the end of the week, I went into the woods behind my mom's house, and there's a
Good news,
spring-fed
everyone.
pond back there. It's this thing, and collected these samples from it, and you can see that the
Nothing
whole
is
surface of this
impossible.
pond is
Not
covered
if
with
you can
duckweed,
imagine it.
and so it's kind of a challenge to get into it and collect the material. Thank you for the birthday wishes. Yeah, it's
Good news,
it's a
everyone!
pond. I mean, I wouldn't swim in it, but you could throw a rock across it. But it, it's It's not quite a lake.
Nothing is impossible.
That's what
Not
we
if you
call a
can
pond.
imagine it.
So I collected some samples from this, and then we were just looking at this pond, which is just
Nothing
like on
is
the other
impossible.
outside of the
Not if
woods.
you can imagine it.
And I grew up swimming in this pond, so and I'd never looked at the diatoms in it either. So I thought, well, I'm here, I might as
Good
well
news,
collect
everyone.
some, and that's what we did. I used an ice cream bucket, and I did have some other tools. My mom lent me her turkey baster. That's okay. I don't really need notification. I know that I got raided, Claire. It's fine. I used my mom's turkey baster, and my mom gave me also a like a salad bowl mixing spoon, so I could kind of scoop some mud up, and then I had an ice cream bucket, like a 1-gallon ice cream bucket that I tied to it, a rope to it, and then I put some rocks in the bottom of the bucket so I could get a good swing, and then I threw it out into the middle of the pond and pulled it back to shore, and here we are, diatoms that I collected. I was in the lab all day today processing these, so I process and prepare them. And then I also got some samples from Pacific Plankton about 2 weeks ago, and so I was preparing those as well. And we looked at some of those on the scanning electron microscope. So if you think we're close, looking at the images here at around 3,000 times, you should see them in a scanning electron microscope, which is where we were earlier today, where we were looking at Pacific Plankton samples from Mountain Lake. And I have some of those also prepared slides here from those from Mountain Lake and also from San Francisco Bay. This is why you don't drink pond water. Well, diatoms won't hurt you. In fact, diatoms are often used as filters. They're in beer filter and wine filter, so they're not going to hurt you. The other things that are in the pond that eat the diatoms probably won't hurt you either, but they would gross you out if you saw them at their full size. There's a nice big eunocia. So people
Nothing
always
is impossible.
get
Not if
worried
you can
about
imagine it.
eating, drinking. The diatoms are the one things you probably don't need to worry about, at least in lake settings. You shouldn't be drinking seawater anyway, but there are some diatoms in the ocean that could potentially cause problems for you, just all belonging to one group. Ooh, there's a cool diatom. It's a little pinularia. It's a triungulate. I can get zoomed in on that and take a look at it. That's a cool one. Very wavy. Yeah, that's the microscope that I'm using right here. And let's see. The camera that I'm using is, so let me see, I can show you everything here. That's the microscope. That's the DM2500. And it's got a camera mounted into a photo tube at the top. And that's just basically hooked to a camera capture card that comes into my my computer. So I use so much diatomaceous earth in the garden. Yeah. In fact, my wife, we had some sort of ant problem. My wife just was grabbing some of our diatomaceous earth that we got from the hardware store that is actually, by my estimate, 10 million year old diatomite, based on what I saw in it, to kill ants or keep them from getting into something, our house maybe, I don't know. When I was younger, I worked at a factory that produced oils, and we used diatoms in a big filter press to take out any water left in the oil. Oh yeah, because they have so much surface area, basically, they can be used for all kinds of things. In the silica gel packs, in hand warmers, in dynamite, actually. So if you look up some of the uses for diatomite or diatomaceous earth, if you like, there's a whole bunch. And of course, the living diatoms are also beneficial to us. that is a chrysophyte cyst right there. That thing, that's actually not a diatom, but is a chrysophyte cyst. They look like Christmas ornaments. You can see it's got like a stem. There's a stem right here, like a little V-shaped stem. And then it's just basically a globe. It's the stomatocyst, so it's a way for the organism, the chrysophyte, to hide in conditions that are unfavorable. So, Steve Mandel asked, if I buy diatomaceous earth, can I find fossil diatoms in there? You better be able to. That's that's what it should be composed of. Most of it that I've found is 10 million years old, or between 10 and 6 million years old, and it's usually terrestrial because those are the easiest ones to get. Yeah, you can find intact ones. I put it right on the SEM, in fact. And you can just sprinkle some of it onto a cover slip or a slide and add water or whatever you're using to mount, and you can just look at it right away. We did that for one of our streams. I sprinkled some on an SEM stub, and we just put it right into the SEM. Some of them are broken, but a lot of them are just completely intact. Yes, it's the same mountain lake, yeah. So that's not this sample, but I will put that on here in a second. Hey, Devil and Mrs. J. There was a question, Henny Swarwerp said, do diatoms make oxygen? They do. Estimates range between 20 and 40% of all oxygen on planet Earth coming from diatoms. So effectively 1 in every 3 or 4 breaths that you take is oxygen that just comes from diatoms and nothing else. They're found in the oceans, they're found in rivers, they're found in lakes. They're the primary producers. They're the grass, if you like, of lakes and in the ocean. And everything eats them. So that's, they're the foundation of the food web, and pretty cool in that sense. You can see some of those organisms getting eaten if you'd like on Pacific Plankton stream. She should be streaming tomorrow, maybe, if she gets back from Tahoe by then. But this is a Celophora. That's a diatom right here. There's 2 valves, so if I focus through, you can see the other valve. It's one of the things that's cool about a light microscope is it's sort of like an X-ray machine in the sense that the light comes from the bottom through the stage and then up into your eye. So everything you're seeing through it. And I can focus on different planes, even ones that are below the surface. So that's the valve that's on the bottom. And if I continually focus up, you'll see it come back into focus, and that's the valve that's on the top. So we can actually see both valves of the diatom pretty easily that way. You used to take your students there all the time. Well, you probably got some of those diatoms on your, I don't know, swim clothes or something. Sandals? I don't know what you wore out to take your students out there paleontologizing. Let's take a look at some of those samples because they're pretty cool. And that's what we were looking at today on the SEM as well, some material from Mountain Lake. So diatoms are nature's Skittles is what you're saying. Yeah, taste the rainbow. Chops, taste the rainbow. Yeah, breathe the rainbow, I guess I should say, yeah. On your boots, okay. I don't know what you wear in the field, who knows? All right, so this sample was, that's one from my mom's pond, and I'm effectively calling that Diamom Pond, which is my mom, and she's going to watch the stream later, so if you want to say hi to my mom, now's a good time to do it, because she's going to probably stop watching when I get to the part about California lakes. So this next sample is one from Mountain Lake in California, and let's see, there was a lot of material on these, and I looked at the unprocessed and the processed samples, and I didn't really notice any difference between them, other than there's a lot of junk on the unprocessed ones, so we'll just skip those and looked at the processed samples. So these were rinsed in nitric acid for on a hot plate. I don't know if that's rinsed is the right word. Boiled in nitric acid for about an hour and a half with some hydrogen peroxide added to get rid of all the organic matter in them. And then rinsed afterwards to get rid of all the acids and the oxidizers, so that it was just neutralized. And then I just took a couple of drops of the remaining extract and dropped those onto a cover slip and let it dry, evaporated on a slide warmer, and then on a hot plate where it was cured with a resin. This is Nafrax, which is a special resin that lets us see diatoms more clearly in the light microscope. So this is what we're looking at. There's a lot of diatoms in Mountain Lake, and almost everything here, these things are all little colonial fregillarias, fregillariales. These are probably Starosiras, all of them. There's a lot of them, and they form chains. So each one of these little boxes like that and that's one cell, that's one cell, that's one cell with 2 valves each, and that's one cell. And there's 4 of them living in a colony here, and between each of them there's little linking spines that sort of like knuckle together so that the valves stay together. And you can see they can have much longer colonies here. And this is a amphora, it's a different diatom, but you can see that there's the whole frustule is present. There's one valve, and there's the other valve. And these were collected by Pacific Plankton, who's here as my moderator in the channel. And she's got her own stream, which you should check out. And someone could probably just type !PAC, P-A-C. And then there's all kinds of information about Pacific Plankton streams, but she streams usually on Tuesdays and Thursdays. And that's my main job in life, is to be her moderator, and then also I'm a professor. So just in my spare time, I'm a professor. So I encourage you all to give her a follow. She's does live streams from San Francisco Bay, and spectacular microscope stuff. That is a Starosira. I don't know what the species is, and I know most of the Starosira. I'm not sure what this one is, so we might need to look into what this is and potentially a lot more closely, because it doesn't fit anything that I recognize. There was also, we saw this sample when it was live in Pacific Plankton stream. Like I said, it was from 2 weeks ago. This is from June 15th. That big guy right there is, I'll zoom out. Now the scale bar is correct. That is Cimbella mexicana, maybe some variety of which. It has a stigma here in the center, and then it's sort of this banana shape. And you can see that the striae are kind of boxy, but we've seen this in the scanning electron microscope. Let me see if I can find that real quick for you. I ought to be able to find my own SEM images. Just so you can get a sense of what the little boxy things you're seeing are. So on the outside of those boxy things, is it this one? Oh. Let's see if I get rid of those. Not showing. Oh, it's probably just the wrong box. It doesn't want me to show that. Well, we'll get back to that later. If you just go to the Instagram account, it's like the second image back. It's actually pretty recent. There. The top one, not the bottom one. It's 3 back. Yeah. It's sort of gold colored. You can see the structure of the actual striae, which are really sort of spectacular in the SEM. Oh, I guess I could have just given you the whole link. Now you don't have to search. For this guy, there's some varieties of Cimbella mexicana, but it's one of those. And we saw something really cool in here when Pacific Plankton was streaming that got me kind of curious, which is, those are Cyclostephanos. Bunch of Cyclostephanos. Aroicosphenia, which usually live in flowing water environments. And it didn't look like any Aroicosphenia that I know. So, again, it's probably something unusual. There's Calanice amphispana. That's a cool diatom. Used to be my wife's favorite diatom. We saw one of those on the SEM today. We were kind of poking around in the same sample from Mountain Lake. It's a Nitzia. Some more Nitzia in between all these Staracyra. There's a little Navicula. Some pretty cool diatoms in here. I can hear fireworks going off still. That is a Triblinella right there. I'm not sure which one. Let's zoom in on it. You can sort of see it's bean-shaped or snow pea-shaped. And then Triblinella has a really characteristic, like there's no striae and it's kind of curved on the valve face, and you can only see it when you focus through it. So there's a part here that's out of focus constantly, and that's a characteristic of Triblinella. That one we probably could find pretty easily on that. It might be on Diatoms of North America. Let's keep looking around in these samples. Thank you for all the follows. I'm sorry I didn't catch all the names. It's complicated to run a light microscope and carry on a conversation with chat at the same time because my eyes are usually stuck in the actual instrument over here. And, oh, that's another thing that we saw a lot of. This is a rather unusual diatom. This monster here is a terpsino, and the ends are broken out. I keep finding them with the ends all busted out. That's a really pretty diatom. So this sort of wavy structure, and then it has these dark bands that are running across the middle. These are costi, and then usually on the ends it has what are called ocelli here and here, which is just a bunch of little tiny holes which kind of look cool. And I tried to get one in focus for us today on the
Good news,
SEM.
everyone.
Failed, generally because they were all broken. And let's see if I zoom in, right there, you can see the valve face. All these little dots that are on here on the surface. And then this is sort of like a blank area, a highline area in here with this narrow elliptical shape. That's a Rumuportula. That's the external expression of the Rumuportula, which is a feature that should exist on all Terpsino. And you can see it's right here in the center of the valve. And this diatom is very likely Terpsino musica. I did see some Girdleview images of it, and in Girdleview, the diatom has a what looked to be little musical notes created by the Costi. So we should go check that out. If I find it, I'll try to zoom in on it. That's a Samothidium of some type. There's a bunch of these giant Cerarella that were taking up most of my SEM images, actually, because I was kind of focused on the big things. There's a Nidium. There's a little Cockneys. Here's another Tribulionella. You can really see the way the central sort of line down the middle doesn't have any, it's sort of like a blank spot again. That's really characteristic of Tribulionella. Like, it's not in focus when the rest of this is in focus because it doesn't have any striae that run through it. You can see right there. And that actually runs the length of this sort of circus peanut shaped diatom. Hey, Brendan, can you give a shout out to Brendan? Oh, you did already. Okay, good. And also, we can't play Hannah. I can't play any songs by Hannah or Rebecca, but we can give her a shout out. And also, I have this. If you want to hear songs about about my SEM stream and sung by a musician here on Twitch, there's a link right here that goes to it, and she wrote a whole song about my SEM stream, which I found absolutely amazing that somebody would take the time to do that. But you should check her out. She's a great musician, and Brendan is her primary moderator, so next to her significant other, Radio Joe. And one of my favorite streamers to watch on Twitch. So I'm almost always in her streams, hanging out in chat whenever she's there. And I think Pacific Plankton has now also become addicted to it. So very fun. And it's a Epithemia gibba. Here's that Monoraphid diatom. I think it's Samothidium, but I'm not positive about that. Here's a Diplanese. If you haven't guessed, there's a lot of different genera of diatoms, like hundreds of genera of diatoms, which makes their taxonomy somewhat troubling for people to learn initially. And Pacific Plankton was in a class this summer, an online class to learn the diatom genera and to learn how to identify them to species, taught by some of my colleagues. And so, getting close to the edge of the slide, it's acting weird. That's a Epithemia. That's a good one to zoom in on. You can kind of see a lot of detail. I think both valves are there, which makes it also a little bit glowy like this. But these dark bands that make it sort of zebra stripes are Costi. This line that runs along here, this dark line that then hooks up towards the center right here, is the Raphi, which is how the diatoms crawl around. And I did say crawl around. Diatoms with a Raphi are actually motile. They can crawl and get from one place to another. So it's pretty cool. Sideways contact lens. Yeah, some of them actually do look a little bit like contact lenses. Yeah, there's an official stream theme song. She's going to record it for me, and then I'm going to try to put it to a video one of these days. This is sort of like a little banana. I like to think of this one as a fly's head. It has a sort of like bug eye texture to it. And these actually look really cool. See all these little sort of dots? Those are the areoli on the valve face. And you can count those and the concentration or the density of them to help you figure out what the species is. I'm pretty sure it's Epithemia adnata, but you can check my species identification easily by going to the link that Pacific Plankton provided for Epithemia earlier. That's the Reucosphenia that I'm kind of wondering about, but we didn't see many of it clearly in the SEM because I was kind of running through looking at all the samples a little bit. So we'll either look at it on Wednesday or maybe next Monday and try to look at these samples again a little bit more closely. And I might actually sneak on the SEM tomorrow and do a little bit of work on these as well and see if I can find those Reucosphenia. It's really strangely shaped. How do they crawl? Steve, they crawl sort of like a snail does. They have that sort of slit that runs through the middle of the valve or sometimes through the edge of the valve called a raphe, and then kind of semi-pneumatically like a snail's crawl. This is Cyclostephanos dubius right there. That's what that is. But they sort of crawl around using that raphe as their motility tool, drag themselves around. I don't know. The mechanics of it are still sort of baffling to me, but they're single-celled organisms, so I think it's kind of cool that they can kind of move without any sort of flagella or cilia even, which is how most things that are little like this get around. That's a different epithemia right there. That's Sorex. That looks like maybe it's a Basilaria. What do you think, Pacific? Get some Basilaria in my sample? That's your sample? That's what it looks like. It's running right down the middle of the valve. Fancy pen? You know what? When I start with my students and they're trying to learn the diatoms, I don't want them to be intimidated by the names, because it's less important that you can tell what the name of something is, and more important that you can regularly identify it as a distinct organism. And so I just tell them to give them their own names. Like, just use whatever you want, like fancy pen. That's a perfect example. And then later on, when they've seen them enough and they're sure that fancy pen is always fancy pen, it's always the same one, then I sit down with them and we sort of go over what it could be and put a name on it, the real name. And they can still call it fancy pen for as long as they like, but if we go to publish it in a paper, fancy pen probably won't work. So this is another banana-shaped diatom. That's a Cymbella. It's named that for it's shaped something like a symbol. You have to imagine it flipped over. For me, it's in the microscope, it's actually flipped over the other way. But the whole thing kind of looks like a cymbal, like you play on a hi-hat on a drum. And so it has a distinct shape. They're all kind of that shape. Sheer force of their own willpower. Some strong willpower in the diatoms. So I try to get them to give their own, yeah, fancy McFancy pants, whatever you want to call it, just so long as you know what it is and that you always call it the same thing. Hi-hat, yeah, exactly. It's a cymbal. It's spelled differently than cymbal, but I think that's where the word comes from. We saw a Salafra, that's what this one is, in the other sample. It was a different species, but it's the same genus as this one. So you're getting a ton of diatoms right now because I'm just looking through a sample that was prepared for diatoms. This is the way I like my samples to look, which is full of diatoms so that I can count them and identify them in the opposite order to that. But that's Alicocera granulata. Most of these I already know, or many of these I already know to species, but there's some in here I would still have to look up. So there's a cool little Diplanese. I don't know what that one is. I know it's a Diplanese. I can get it to genus. There's something like 8 or 9,000 species of diatoms that have been recorded in North America, and I'm sure that number is way lower than the actual number. So I also try to explain to them that they're never going to memorize all of the diatoms, that eventually, they're going to have to just realize that there's some that they won't know when they see it to species. But normally I can tell you if it's something that I've seen before. I can usually go, oh, I know that one. It's this thing. And if I can't remember the name of it immediately and I've seen it before, I usually can I can find it where it is in the book pretty easily because I know where it belongs in the book. Usually I spend some time looking through books to try to find
Nothing
the
is
diatom's
impossible.
name.
Not if you can imagine it.
Then I usually can figure out I usually remember its names, which is funny because I'm terrible with people names. Apologies to people. But when I meet somebody and they tell me their name, I almost always am doing, like, my brain is trying to do something else and like process who they are or something. And I almost never remember people's names the first time. Like I have to have somebody tell me their name after I've had a chance to like see them for a little bit, and then I usually remember their names. But diatom names I'm pretty good with. It may be that I like diatoms more than people, but so for the ones I live with, I kind of like them a lot. That's a Pinularia, by the way. I don't know what species because there's too many Pinularia that look too similar, and I usually have to do a bunch of analysis on it to figure out which one it is. So most of this material is tychoplankton. That's what these little guys are. They look like zippers or boxes all together. And they usually indicate intermediate water depth. They're kind of weedy species. They're found in all kinds of environments. And then the rest of these are mostly benthic. And the only true plankton in the samples that I've seen are the little round guys, which are like this one. That's a Discostella stylidura, and that one is Cyclostephanos dubius. And the Alicocera granulata. And I don't see any other that's like truly plankton, but I'm pretty sure these samples were collected with a plankton toe, like a plankton net. You can correct me if I'm wrong about that, Pacific, but I think she collected them with an actual plankton net, and the rest of the stuff just kind of got kicked up, probably by wave energy. Mostly it's these sort of heavy benthic stuff that live in tube colonies, or they live attached to substrates. Like this thing right here, Triblinella is edaphic, usually found in soils or along riverbanks. This thing here is epithemia. That's probably living on plants in the shore. The rest of these guys are tychoplankton. They're usually found attached to some substrate. So I don't just look at these things, I actually know a lot about their ecology as groups. And I use them to try to figure out what was going on in lakes. And this guy right here, Cyclostephanos dubius, suggests that the lake has relatively high phosphorus. The presence of Oligocyra granulata also suggests that it's relatively high in both silica and phosphorus. And the other one, Discostella stolidura, the little tiny one, indicates that it probably stratifies in the summer and seasonally is probably abundant in the lake. The rest of these guys are little benthic stuff for the most part, which are just living on the muddy surfaces or plants. And so they're hard to provide much information for us, except for there's a lot of these tychoplankton. So there's a lot of intermediate depths. And then there's some salty stuff in here, or potentially springy stuff in here, like sort of things that you'd find where there's a freshwater spring, or potentially like hard water spring. So that's the terpsino, the big terpsinos that we keep seeing, and some other things in here that I thought were sort of salinity indicators or slightly saline indicators. I saw a really cool diplonese that looked like a marine diplonese, and I don't know if those are being blown in on the wind or carried in by birds. A lot of them look a lot more ragged than the rest of the diatoms too. So there's a cool one. The Gyrosigma. You can see it's got a little grid of striae. They're not diamond shaped, they're arranged in a sort of a lateral grid. So yeah, there was a Ricusphenia back there. And oh, you didn't use a net. Oh, how did you collect these? It's a spring-fed lake. Look at that. On a flyway. I'm pretty good at figuring out the diatoms, what they indicate to me. You set up a green screen and have Hannah play a diatoms song in front of this. That would be great. Oh, you scraped them from a rock, or oh, with a turkey baster. Okay, like from the surface of the mud, because a lot of these are epipellic. Like those big Cervarellas are probably epipellic, and this thing right here, this Gyrosigma, is also epipellic, meaning they grow on muddy surfaces. So that doesn't surprise me either. Okay, well, we've looked sort of through this for a while. There's a bunch of other cool stuff in here around muds and rocks, yeah. So I feel like we got a handle on the types of organisms that we see in this particular sample. I made some extra slides of this for you, Pacific Plankton, and I will send them your way. So I have a couple of slides from Mountain Lake for you, some unprocessed material from Mountain Lake, and then this material from the actual San Francisco Bay here as well, from this collection that you did on June 15th. And we'll look at some of the live stuff first. It's not actually living, but it's unprocessed, so I just like to call it live because it still has some of the living stuff in it. We might see some things that aren't diatoms in here, is what I'm saying, because they haven't been processed for diatoms. They're just, whatever was in the sample. We rinsed out the salt water, and then and some of the mud, and then look at that, there's the Cimbella mexicana right there. and then whatever else was in the sample just ends up on here. So I probably need to zoom out, actually. It's a Melisira. Let's go at least out to this. These are sorts of diatoms that we see all the time on Pacific plankton streams. That is Actinocyclus. That one is Melisira. Holy crap, that's a big colony. That thing is monster sized. That's an odontelloid of some type, right? I think we looked this up before. The stinky ones. De-enhance. You need to print something? From this computer? From this computer?
From anything that can print to that.
Well, I don't know what prints to that.
Yeah.
Good news, everyone!
Do you need it printed right now?
I can't do my work until I have the labels printed. So.
Oh, okay.
I tried to add my laptop only sees printers on the university network. I can't figure out how to get it to see.
Do you have the file? I probably can print it.
Good news,
My
everyone!
wife needs something printed. Yeah, just bring it and I'll print it. Shouldn't be a big deal. What is it you call these all the time? Tropidinese or something like that? Yeah. Exactly. Look how huge that thing is at 1,000 times. Are those living diatoms in water? Cargo cult, these are the these are unprocessed materials. The diatoms are, however, mounted. These are prepared slides. So the samples are basically cover slips that are epoxied onto the glass slide, and everything that's in there is frozen in time. So at one point they were living, and then they got shipped to me, and then they sat around in vials that got the salt water rinsed out of them for a couple of weeks, and then I dried them onto a cover slip. So you might see some chloroplasts, like the green stuff that you see in there is probably like in here. Those are probably like ruptured chloroplasts and original material that used to be part of the living organisms when they were alive, but they're not alive now. So nothing in here should move. It's been locked into place using that resin, and that's a ditillum, and that is a Thalassocera. You can see there's some green color in that one. Those are actual chloroplasts again. They're just locked in this epoxy, so they're basically just frozen in place. Yes, there's a Raytheon Tropidinese. And I don't think Tropidinese is its modern name, but we had that conversation before. Yeah, I think I need to zoom out because we can't see anything. We're so close. So I'm going to undo that. Is that your glasses? Alright, let's zoom out a little bit. Sorry for the brightness. This one?
First 4 pages.
Not the whole thing?
Yeah. Well, the 5th page is incomplete, so it would just be a waste.
Solving problems for my wife. Plagiotropus, yeah, that's its modern name. Alright. Should be solved. Yeah, there's a big colony of them. What is this one? I think it starts with a D. It's not the Tillum. Oh, what is it we call those? Yeah, there's some Campylodiscus in here. I can't remember the name of that genus. Look, its printer is shaking my whole... It's like an earthquake.
One more page.
There's some excitement. It's like they're moving, but they're not. There's a little colony of those Socialis, Lesiceras. And that's a Ellerbeckia sole. Alright. Crisis averted. Now she can work. There's a diatom on its side and a Cattosaurus. The printer is now adjusting itself. There's a big Cossinodiscus. We saw this earlier on the stream. I took a picture of one of these things. Let's go... What are we in, 40? Let's go zooming in at 40 here. So we saw one of these that was identical to this on the stream earlier today. It has these little like hexagons with dots in the centers. Lithmodesmian, yeah. It's the Desmian part I was remembering. Do I study living diatoms under a microscope? Yeah, on occasion I look at the living ones. I look at all kinds of pond organisms occasionally that are living. My primary focus is... Oh, there's a hairy shoulders guy. It's a different one than the one we saw. Look how huge that is. Monster. It's an absolute monster. It's a little baby. And then look at all the detail when you zoom in. It's got the hairy shoulders. We've seen those hairy shoulders on the SCM. Do I like the dead more? I like diatoms that aren't moving because it's easier to identify them. And also, when they're not all full of chloroplasts, they're easier to identify for me. Do I like the dead ones more? I mean, I use them for paleoecology, so my primary occupation is reconstructing past water environments. You know, I need to know a little bit about the modern living diatoms, and then also I need to know what do they indicate. So it's sort of a combination. You know, it's helpful to see both, basically, is what I'm saying. There's a whole little bunch of them together too. Those ones that you see living in a little group. Those are all little diatoms. They live in a colony together. A colony filled with like mush. There's a Trapides or Plagiotropis and some Thalassocyra. Earlier today we saw a really gorgeous Arachnodiscus, and I'm pretty sure there's some Arachnodiscus in these mounted samples as well. Just require me to look around quite a bit because they're kind of uncommon. Seeing a bunch of cool little round guys. That's a tintinid skeleton test. There's another one. You can see it's glued together, all of these little particles, and some of them, like that one right there, that little puzzle piece, is a diatom piece. That's a fragment of a diatom. And most of these are just pieces of silica, probably silt. And then this is where the organism comes out. It usually has like little tentacle-like hairs that stick out there. Did you find a Mona Lisa of diatoms yet? I don't know about a Mona Lisa. Hey Savory Pancake, how's it going? I still remember you, even though it's been a long time. That's an Actinocyclus. In the SEM, these things have big Rimoportula that look like ears. We saw one of these today, usually located right here at the end of these rays. There's these big ear-like structures that are the Rimoportula, and we saw some of those. There's an Actinopticus. This is the radiation symbol diatom. If you are a subscriber or you want to use the emote, that one is there. That's Actinopticus.
Good news, everyone!
And also this one. Oops. Those guys. I'm going to observe, but I do have a question, but I don't want to interrupt what's going on. Oh, there's nothing important going on. I'm just looking around and, you know, doing the normal sort of thing I do. Looking at diatoms from wherever. Trying to figure out what I'm looking at. That's a Thalassocyra. It's got that really pretty blue color in it. That's just a physical coloration. It's not real. This is a result of refraction of light as it passes through the little holes in the diatom. But you can see they're important for us for identification because you can see these sort of rays coming out. Like here's a triangle shape, and here's another one, and here's another one. And they tell us that this diatom has sectors. So it's divided into little sectors. And each one of those sectors, sort of pie-shaped sectors, that's a common feature for Thalassocyra. Also, it usually has a row of Mantophotoportula around the outside edge. That's another characteristic to let us know that we're looking at Thalassocyra. And somewhere on here is a Rimoportula, which is a really long spine that's sticking up. So, yeah, oh, there's an orange one too, I forgot. Yeah, you can go get the orange one. It's on the BTTV. So there's a cool Cossinodiscus perifera. We saw one of those today as well. A lot of the same diatoms that we've seen on the stream because we were looking at a lot of the big round guys on the stream, on the SEM stream earlier. And that's a little Cyclotella that's, because we're looking at a bay sample. Mixed environment. I'm really happy that Paleontologizing brought some of his people in. I hope some of them stuck around and enjoyed it. And that's a Paralia, stack of Paralia, I think. Probably a bunch of them have been here before because he's raided me a bunch of times. There's a Plagiotrophus. You're asking about the Rafie for these pack, and the Rafie actually runs right along this edge. That's actually the girdle view, so it's like a keel that stands up off the top of the diatom. Okay, I've been into personal organic gardening and I've been told to use diatomaceous earth for insects. Do you know much about this? If so, is it harmful to beneficial insects as well? Is it comprised of only diatoms? I don't know how it works, and all my Googling says it just does. I'm not the best at search queues. So I actually talked up a little bit about this earlier. Yeah, they don't know the difference between the good bugs and the bad bugs. It's not a keeled Rafie. It's like running through the axis of the valve face, but we can't see the valve face because it's standing on a long sort of bumpy surface here. So if this is the girdle view, so you can see what the valve face is doing, and we almost always see it in girdle view as a result. Okay, so to answer your question, how does diatomaceous earth work? Generally it works like this. Diatoms are very small, they're made out of silica, and diatomaceous earth is usually made of, fragments and whole diatoms. And the little bits of silica are very sharp, and when insects walk across it, it's painful, for lack of a better word, or it cuts into them. And so think of it like walking across the gravel driveway. Like, it's just painful, and so effectively they won't walk through it, or if they do, it's a struggle for them. So any organisms that crawl or walk across surfaces where you're sprinkling caltrops, basically, if you're familiar with that concept, sharp fragments that they have to walk across or glide across. So snails, slugs, any insect that walks, like ants that don't typically fly, are going to be affected by it. It's basically glass. Yeah, it's opaline silica, so it is basically glass. And it's like shards of glass on the ground that they have to walk over. And most of the organisms will not walk across it, or they will and they'll get hurt. That is a Campylodiscus right there. And a Tintinid, Actinopticus. There's a Nitsia sigmoidea in the background. Cocinodiscus, Lasicyra. It's a nice little bundle of diatoms right there. Most of the things. There's some more of that physical coloring. There's some more of that physical coloring. That's Actinocyclus right there. And I colored one from an SEM image that I tried to make it look kind of like this rainbowy color, because this is what they look like at 40x from the SEM images. Yeah. So, caltrops, exactly. I don't know if they actually breathe it in. Mind over matter insects can walk over it. Possibly.
Good news,
Sam.
everyone.
Chicken is nice. Alright, if you say so. Thanks for the follow. I think ants would be more affected than hornets, yes. I do like hornets for their killing of moths and whatnot. I do invite predatory insects into my garden. I know hornets don't live that long, they're just feeding their young. Yeah, it depends on the type of wasp or hornet. I don't know about, I like wasps, I don't know about hornets, but I know a lot more about them. I'm just watching this while sleeping, so kind of ghosting. You can watch stuff while you sleep? That's incredible. Basic hornet. You should teach me how to watch stuff while you sleep. That sounds really cool. If you lurk, that's totally fine. All lurkers are welcome. I'm assuming you mean you're just listening to it, which is also fine. I know a lot of people seem to like my presentation style. I try to keep things calm and pretty chill in my streams. All right, let's find another slide to look through. How about that? We can look through some more of this material. That's Pseudonychia right there, that little group. It's a little Thalassocera, some more Pseudonychia, Agnanthes, Datylum, Actinopticus. Oh, there is a skeletonema. I didn't see any of these in the stream. That's a pretty one. Caught a mostly completely in focus Actinopticus right there. No funny angles or junk on it. You can see the different sectors. It's got one elevation, one set of depressions that are next to each other. And people usually refer to this as the radiation diatom for reasons that should be clear. Dreamstream. Yeah, the radioactive sign is exactly it. Actinopticus. Oh, it's not radioactive. As far as I know, there aren't any radioactive diatoms, but they did find some diatoms that were in the cooling towers of... there's your Plagiotropus right there. Both two sets of valves, recently split. I did find some. There's an amphora for indubitably Brendan, or Hannah Rebecca Music sings about amphora in their song. It's Cuccanese. It's one of these really pretty Actinocyclus again. This little bare patch right there is a special process on these called a pseudonodulus, another characteristic of Actinocyclus. They found some diatoms that were in a cooling tower for a nuclear power plant, and they actually got their own genus. They were so messed up, but I don't think they were radioactive in the sense that they gave off radioactivity. Just tell you if you find anything you might want to see. I'm not sure what you want to see. You know, there's cool stuff everywhere in here, but you know, I don't want to wake you up. If you feel like you're getting to sleep, just about to fall asleep, then fall asleep by all means. What time is it? It's almost midnight Eastern, so pretty soon I'm gonna want to go to sleep. Been streaming for a while. There's a long chain. Pseudonychia. Coscinodiscus. That's an amphora. Coscinodiscus. A bunch of big Coscinodiscus in here. There's a piece of a copepod skeleton. Alright, let's switch from this live material, or once living material, over to a sample that's been processed for diatoms, because it will get rid of all of this debris and also some of this green stuff and leave us with just the diatoms. Oh, that's a pretty pretty one. Look at that. Look at the color in that. That's all physical color. Like a rainbow. We can actually... That's as close as I can get without going to the 1000x objective, without sort of blurring as we get closer. 3 times is probably the best. It's a pretty color. Set of colors. A rainbow diatom. Have we already studied diatoms from another planet? Polar bear, I don't think there are any diatoms on other planets. I think they're exclusively Earth beings. Organisms. But if somebody wants to get some diatoms from another planet and send them to me, I'll absolutely study those. I look forward to it, in fact. Be excited to see it. People have found some things they thought were diatoms on meteorites that they thought were brought in on the meteorite, but in fact I think they're just contamination, because it was things like Nitzia. Like, if I can identify it to genus, it's probably not from outer space. Okay, let's switch really quickly to another one. This is the same material, it's just been processed. We'll zoom out to 10x from 40. Oops. Hello, brightness. And we'll just look around at 10x and see what we can find. As you can see, the samples are a lot more diatom rich when we got rid of all the other junk. Unfortunately, it also gets rid of some of the diatoms. See that one that's like really blue colored? Let's zoom in and look at it. Well, it's like purple when you zoom in one level. Let's see, 40x, what does it look like? Still has sort of a purple coloration. It'll look better when I put the right settings on it. It's also actinocyclus, I'm pretty sure. There's another pseudonodulus on that one. So it's basically the same, just it doesn't have as much of the rainbow pattern to it. It looks more like red here. But again, it's not real color. It's a really pretty diatom though. Nitzia. It's a piece of Catoceros. Pseudonitzia by itself. Some more Catoceros. Plagiotropis is everywhere in these samples. Here's another one. Oh. There's 2 of them that have their rainbow colors. Side by side rainbows. Oops. That rainbow is probably largely because I have polarized light. I wonder what happens if I turn it a little. There, you can see what happens with the polarizer. The maximum color comes at the highest angle. It's right at 90 now. I'm definitely going to watch you more. Good. Great. Are they spherical or flat? These ones are sort of like slightly domed, especially around the edges, but the valve faces are pretty much flat. And that's a pretty common pattern. Like these ones are undulate. That one is very flat, like a pancake almost. And usually you can only tell by how much of the diatom is in focus at the same time. Those ones have a slight dome to them. Those ones are undulate. Again, that's pretty flat. Most of the Cockneys, or not Cockneys, the custom discus are pretty flat. That one's got a dome. You can tell from the way that I can only get the center in focus and then the edges. So we can use our optical dissection to sort of see whether they're flat or rounded. None of them are spherical, really. Although these are really domed, or they look really domed anyway. They're still not spherical. At best, they're kind of a low angle dome. They're definitely not like a ball. The ones that are closest to the ball are like Stephanopixis. They're pretty much like a Epcot Center dome. And we've seen those in the SEM, and I think I saw one on the SEM today, and we see them occasionally here in the light microscope as well, and on Pacific plankton streams, of course, frequently. During certain times of the year. They seem like they were around in the spring kind of a lot, and then haven't been around quite so much since then. Still see them occasionally though. That one's got a bit of the rainbow coming out. Let's go up to 20x. Spacey One, hello. Thank you for the raid. Guess what we're looking at? Diatoms. Once again, we're looking at diatoms. Thank you for the raid. How was your stream? Forgot to ask Paleontologizing how his stream was, but his streams are always good, so I know it went great.
Good news, everyone!
Like 150 people. Couldn't have been bad. So we saw a bunch of those weird sectors. We saw a bunch of Arachnodiscus in the SEM today, but kind of sparse in these samples. Although I have seen some when I was looking around earlier. I don't know which slide I was looking in. I did see some Arachnodiscus. But the SEM Arachnodiscus images were just great earlier today. And we'll probably, as I mentioned, we'll probably look at some of that again on Wednesday or maybe next Monday. That's a really pretty blue color that's being created. That's an interesting diatom. That's Actinopticus, but it's one that has many rays. Like the orange one that Devil and Mrs. J put in the channel a little bit ago. It's like that one. So 1, 2, 3, 4, 5, 6, 7 instead of just 3. So let's see. What do we got going on here? You know, they sent out a rover to Mars, but did it come back yet? I don't think it's coming back. I think it's a resident of Mars. Chicken. I'm pretty sure it's a permanent resident of Mars now. thank you for sending out the shout out to Spicy Juan. I love the way some of these look. They all end up in the SEM. Yeah. Umbrella Corp. Kind of. My battery light's flashing. We're getting close to the end of the stream anyhow. Ooh, that's a cool diatom. Kind of confused what's going on there. Oh, I think it has like 2 horns and they're coming at us. I'm not positive about that. That's an interesting one. I think it's a benthic. 2 hours and 4 minutes. I add that together with my 3 hours and however many minutes from earlier today. It's 5 hours streaming. It's a lot. It's a lot of time looking at diatoms. It's almost a whole day. Whole work day. Oof, look at the color on that. Well, these are neat. But, I do think we're getting kind of close to the end here. Probably the next time you'll see me stream will be Wednesday. And there's a diatom. Where I plan on doing a stream from the SEM Wednesday afternoon sometime. There's a piece of an arachnodiscus pack right there. Might be as much as we get. One little fragment for today. And we'll see some more on the SEM on Wednesday probably. And if I do a little bit of work on it tomorrow and get around all the other responsibilities I have, I might stream it into Discord for you if you're around. I don't know, you'll probably be traveling. So, might have to wait till Wednesday. In Valview. It's custom discus. Time Traveler, hello. Let's see. Do I think for $200 I can buy a good microscope for an amateur? Do you recommend a brand? Polar Bear, you can get a decent microscope for $200. That's good enough for an amateur. You know, and if you decide you really like it, or you find yourself doing it a lot, then you can always upgrade to a different, you know, more expensive microscope. I'm gonna see if I can come up with some good suggestions for cheap brands, but it depends on what you're looking at, to be honest. Are you gonna be analyzing diatoms, or are you just gonna be looking at things living in the water? you know, if you're looking for water bearers, you probably only need like a 10x objective or maybe a 20x objective at the most. And you can get any, really literally any reasonably decent microscope would allow you to see that sort of thing clearly. You know, if it's for you or for your kids, something like that, you know, yes. And I don't really even think the brand matters that much at that point. Where the brand matters is when you start to go up the scale. If you start looking at $1,000 microscope, I can make some suggestions, you know, in that realm. But typically, it's not going to matter at the bottom end for what you're trying to do for the low end price range. But join the Discord, or you can join Pacific Plankton's Discord. I think if you just click on her name and give her a follow, and also look at her socials, you'll find a Discord link there. We answered this question a bunch of times together. So, but you can, either one of our channels will sit and chat with you about, you know, what we think is a good microscope versus a, you know, not so great microscope for what you want. You're driving in the afternoon, okay. Let's see. I'm thankful my dad gave me a microscope for like your 10th birthday. Oh, that's cool. If you had Amazon links we could buy from, you could make a little side money. I could try to find some for you. I don't really want to make money off of other people, but you know, I'm willing to look for like good choices for you. I looked at flies and you couldn't eat after that. Flies are kind of crazy. In a stereo microscope, they look kind of good. Oh, there's a roundish diatom right there. Those are Melisairas, living in a colony. Should probably move out so you're not constantly spinning through what I'm looking at. Anyhow, you know, just drop a questions into the Discord, and either Pacific or I will get to it, depending on whose channel you put it in. We'll do our best to try to answer that question for you. All right. Lots of cool stuff on the microscope tonight. Lots of really great diatoms. And some mysteries to solve for Wednesday. At least a couple. So Ellerbeck, yeah, that one looks really cool. Pretty cool with the diffraction around it like that. You're having network issues. Wonder if looking at a butterfly would make them lose luster. I actually think looking at butterfly wings might actually make them even more interesting. They're, where they look like they're furry, they're actually little scales. And I have a moth wing over there that I was looking at on this microscope with my daughter, and I don't think it takes away from it at all. Yeah, the butterfly wings or moth wings, they'll be beautiful in a light microscope, especially a stereo microscope. And you might actually see more color on them, you know, in the same way that these diatoms have apparent color. So, very, very cool. We've looked at them in the scanning electron microscope as well, and I don't think it takes anything away from them. I think they're interesting no matter what microscope you use to look at them. Okay, I think I'm gonna raid someone. So let's see, who should we raid? My choices are art, music, or... Oh, let's go raid Fazarius. She's actually looking at some samples on a microscope. That's a good choice. And if you aren't already following all of these, yeah, that'll be good. All these cool people. Oh, I got beat. I finally got beat by Pacific Plankton. There's a bunch of cool people that use microscopes to stream right there. And we will go raid Fizaria before she finds me and raids me. As soon as I can spell raid correctly and then Fizaria correctly. And we'll hang out there for a little bit before she raids somebody, probably because it's getting close to the same time for her. But she is looking at stuff on a microscope, so a lot of times she's singing, and she's got a great voice. But thanks everybody for hanging out, and for the raiders, all the people who followed. There's a huge list of people to thank for today, for the oops.
Keep gifting out another 5
Spicy Wand for a raid. And we got a raid from Paleontologizing, and earlier a raid from Acellus and Rams Reef. And then tons of people who followed coming in from Paleontologizing's raid as well. And Spicy Wands. So thank all of you. And we'll be back on Wednesday probably. And unless there's a thunderstorm or something tomorrow, then I'm gonna see you then, hopefully. Feel free to drop in. We'll be looking at stuff on the scanning electron microscope that we looked at on the light microscope tonight. All right. Good night, everybody.
A water